Hi, in this video I will talk about the difference between LTP and LTD, difference between long-term potentiation versus long-term depression. Now first I would talk about the experimental schemes that people have done. So most of the recordings, electrophysiological recordings, have been done in mouse brain. Here they cut a coronal section. If you cut a coronal section, it would something look like this, and they would place the coronal section in an electrophysiological rig under a microscope, and here they would put the coronal section and they can supply artificial cerebrospinal fluid which would keep that preparation alive, maintain a physiological condition. Underlying that situation, you can see they can start recording. Here is the recording electrode. Other side, which is not shown, is a stimulating electrode. They can stimulate from one side, they can record the responses from other side. Here you can see a DIC image of the neurons where they are started recording. Now they have stimulating electrode. They can inject current, and they can inject current of different frequencies. They can stimulate at 100 Hz frequency, which is a very high frequency, and underlying this kind of high frequency stimulation, they have found that over time synaptic strength has increased, and the readout is increase in Epsp slope than the baseline. Whereas in case of a low frequency stimulus which lasts for about 10 minutes, prolonged low frequency stimulus can give rise to depression in synaptic strength. So depending upon the frequency of stimulation, there could be different outcomes. One lead to strengthening, other lead to weakening, and there is a bidirectionality to it. Now the question is, what is so special about this stimulation frequency? What is the molecular mechanism underlying this stimulation frequency which can give rise to strengthening or weakening in a context dependent manner? So before that, we should understand what happens just underlying this high frequency or low frequency stimulation. When we give a high frequency stimulus about 100 Hz for 1 second, it would allow calcium entry via NMDA receptor. And the calcium entry via NMDA receptor is drastic. It's dramatic. Huge amount of calcium get in from the NMDA receptor, which would increase the calcium level 10 folds from 100 nanomolar to 1 micromolar. Whereas in case of low frequency stimulation, about 100 Hz for 5 to 15 minutes, people have found that calcium enters slowly, and the resting calcium of 100 nanomolar get increased and maintained at a level of 400 nanomolar to 500 nanomolar, which is not as high as found in case of high frequency stimulation. The high frequency stimulation, the brief and high amount of calcium level has different implication than a prolonged sustained level of calcium. So clearly you can understand that if you give a high frequency stimulation, you could have synaptic strengthening, whereas a low frequency stimulus can give rise to synaptic weakening, both at structural and functional level. Here it's shown at a structural level after LTD protocol, the synaptic size has decreased. Now, what are the molecular mechanisms underlying that? One of the striking molecular mechanism by which the functional alteration happens is by endocytosis of AMPA receptors. Underlying high frequency stimulus, calcium level rise drastically. There are decoder molecules which can decode the calcium spatiotemporal dynamics and can give rise to, the ultimate result is to displaying more and more AMPA receptor to the postsynaptic membrane. Whereas in case of LTD, where you give a low frequency stimulus, the high level of calcium, intermediate level of calcium is maintained for a long duration, and that ultimately give rise to internalization of the AMPA receptors, reducing the AMPA current, thereby reducing the total postsynaptic potential and thereby reducing the synaptic strength. Now here you can see that here is the Epsp. It's a baseline Epsp that you can record. After a high frequency stimulus, you are going to record a boosted response, whereas the scenario is opposite in case of a low frequency stimulation, which give rise to synaptic depression. So clearly, the mystery lies in spatiotemporal dynamics of calcium. A brief and high level of calcium can give rise to potentiation or increase in synaptic strength, whereas a sustained low level of calcium can give rise to depression-like phenomena. The question is how this spatiotemporal dynamics of the calcium is read out inside the cell. So clearly we need decoder molecules which can decode the message inside the calcium dynamics. And two most popular decoder molecules that I would discuss is CAMK2, which can kind of decode the message which is inside the fast calcium dynamics, which last about millisecond and it's an amplitude of 100 nanomolar, 1000 nanomolar, about a micromolar. Whereas a low level of calcium is detected by calcineurin, which is a phosphatase. But how CAMK2, let's just first understand how CAMK2 can read out this kind of dynamics, and after reading calcium, how it can write the plastic changes. So recent experiment by Ryohei Yoshida's lab has shown that with FRET sensors, they have specifically uncaged glutamate into specific synapses, and thereby they can activate specific synapses. A particular synapse they can activate, but the nearby synapse won't be activated. In this scenario, and they have a genetically encoded sensor of CAMK2 activity sitting in the postsynapse. They have found that CAMK2 activity persists for about a minute. And just after CAMK2 activity, there is a correlative increase in the synaptic volume or the spine size. But this scenario is only true when they repetitively give 40 to 50 pulses. A single pulse does not give optimum activity of the CAMK2 and does not translate to a volume change or a functional change. That means a certain high level of calcium is important for CAMK2, optimal CAMK2 activity, and thereby structural and functional changes. Low level of calcium or low frequency stimulus cannot give that necessary drive to activate CAMK2 optimally. Exactly, if we talk about CAMK2, CAMK2 is a dodecamer of 600 kilodalton molecular weight, and CAMK2 has 12 subunits. Each subunit need to get phosphorylated, and this phosphorylation is boosted when calcium and calmodulin bind to CAMK2. But this is not the only process. There are phosphatases which is always trying to remove the phosphate groups from the CAMK2 and thereby inhibiting it. Now there is a competition between kinase and phosphatase activity. Now when there is a huge amount of calcium persisting for very less amount of time, the kinase activity is outcompeting the phosphatase activity and thereby optimally activating CAMK2. Then once CAMK2 is activated, it can give rise to structural change, which is recorded by spine volume increase, and also it can increase the postsynaptic current or postsynaptic potential. And this is restricted to the spine which is stimulated, and this kind of response is not found in a nearby spine. And Yoshida's group shown it beautifully. Now, what exactly happening? Why CAMK2 requires a high level of calcium? It has been shown that CAMK2's Km value is pretty high. That means affinity towards calcium and calmodulin is pretty low. Whereas another phosphatase, calcineurin, has very high affinity, and thereby it has a very low Km. Now, modeling studies have shown that CAMK2 activity persists for long time, whereas calcineurin activity doesn't persist for a long time. Now, we can clearly understand the biophysics of detection. If you have prolonged low level of calcium, it could be detected by calcineurin-like phosphatases, and the kinase activation could be done when there is high and brief and high level of calcium. So, and this biophysics of detection is true across several synapses in several brain regions. But the point lies that how CAMK2 and calcium interacts with other molecular players. So depending upon how they interact with other molecular players, it would give rise to potentiation or depression. Now, we talked about this artificial protocol which can alter the synaptic strength, But the question is whether long-term depression or potentiation-like phenomena is true underlying learning paradigms, or is it the same mechanism used by the brain while they are trying to store or consolidate memory. People have shown that underlying specific inhibitory avoidance training, where mouse is whether a mouse is actually trained to avoid a particular dark box where they get a shock, underlying this kind of training paradigm, people found that certain neurons have elevated their EPSP slope. That means that their postsynaptic strength was potentiated, increased, and that lasted for hours. In case of Drosophila, there are evidences where under aversive conditioning, there is a depression happening in specific synapses in the fly learning center and in between the fly learning center, the mushroom body neurons, and the mushroom body output neurons. People have found that underlying aversive training, which is just a classical conditioning where the fly is trained with order A and given a shock, they are associating this order with shock. Otherwise, they are trained with another order B where the shock is not given. Now only order A and B is given. Now if they have learned their lesson, they would avoid the order A because they know it comes with a shock. Underlying this kind of training, people have found that the relevant circuits, in specific element of the relevant circuit, there is a depression of the synaptic strength. Now it's still unclear that how sometimes synaptic depression or synaptic potentiation can encode exactly memory, but definitely this kind of protocol has good computational advantages, which I have discussed in other videos, and that's why people think that this kind of mechanisms could be used by our brain to encode memory. A recent study by Malinow's lab has shown that brain actually used this kind of LTP and LTD like protocol to encode memory. And they have a simple paradigm. They again did a classical conditioning paradigm, a Pavlovian conditioning, where a mouse is associated, when a mouse is represented to a tone, and while they are given a shock. So the tone is conditioned stimulus and the shock is unconditioned stimulus. Shock is unconditioned stimulus because shock is painful to them. Whenever you give the shock, the mouse would freeze. But next time, if you associate both of them and then you only represent the tone, if the mouse has learned the lesson, it would freeze when the tone is represented alone. And now they know exactly how this tone is actually represented in the brain by the auditory cortex neurons. So they can replace the tone by activating those neurons in the auditory cortex. Now they have an optical conditioned stimulus. Now what they have observed that the underlying conditioning, there is certain increase in AMPA by NMDA ratio, which is a signature of increase in synaptic strength. Not only that, after a paired conditioning where the mouse is trained to pair the shock with the tone, after that, using that optogenetic setup, they give low frequency stimulation, thus applying a LTD protocol, and it's reduced the memory of the fear conditioning. So the mouse would be freezing when it is actually when it has fear, it has a memory of fear, it would freeze, and it won't press a lever which is there in the cabin to deliver food. In normal condition, whenever it presses a lever, it would get a food, but when it is afraid, it won't press the lever. So number of lever pressing is almost zero when it is scared. So this is a readout for its fear memory. But this the fear associated behavioral signature is gone once they have applied an LTD protocol. After one day, they have applied a long-term potentiation protocol, a high-frequency stimulus protocol, and they found that memory came back. Again, they have done an LTD protocol. They saw the mouse is no more freezing when the tone is presented. That means the fear memory is gone. So thereby, just after pairing, they can give long-term potentiation and long-term depression protocol to write and erase memories. But also, their research have shown that LTP or LTD are alone not sufficient to evoke the memory. That means LTP and LTD kind of protocol can give rise to plastic changes at the cellular level, but might not be might not LTP and LTD protocol can encode memory, and it requires a pairing event. After that, LTP or LTD protocol can give rise to memory-like changes. So that also argues that LTP-like protocol could be one of the features by which brain can encode memory. That could be underlying memory, but LTP protocol is not exactly memory. But still, these areas are debated, and more research is required to reveal the truth. So if you like my video, please subscribe, give it a quick thumbs up, and don't forget to share. Thank you.